Amylase. Amylase is discovered and isolated by Anselm Pavin in 1833. It hydrolyzes the starch into the sugar and it is present on the saliva of human and some other mammals where it begins the chemical process of digestion. Amylase have a primary structure with a 496 amino acid and in secondary structure it has the alpha coil and beta surface where that is represented here as a pink color and green color respectively. Tertiary structure of the amylase gives the ability to perform its role in break down the starch and glycogen molecule. The tertiary structure is formed from the several sulfur bond and it is also called as a disulfide bond. This enzyme chain have three domains A, B and C. These domains are generally found on all alpha amylase enzymes. The A domain consists of core structure with a beta by alpha 8 parallel and the B domain consists of the seat of 4 anti-parallel beta strands. Long chain are observed between the B strand located within the B domain is binding site for the calcium sodium calcium that is represented here as a 3-ball. Domain C consists of 8 beta strand is assembled into the globular unit. It also holds the third calcium binding site association with the domain A. Now let's come to the alpha amylase. Alpha amylase is a calcium metalloenzyme. It breaks down long chain saccharide into the maltose and maltose from amylose. Alpha amylase is the enzyme that acts as a catalyst for the hydrolysis of alpha linked polysaccharide into the alpha anomeric product. It has the chlorine and calcium atom that accelerate the process of decomposition of the substance. Amylase enzyme have a double displacement reaction. In first step, aspartic acid acts as a nucleophile and attack to the anomeric center of the carbohydrate and form the intermediate. In the second step, the glutamic acid acts as a base and takes a proton from the water and that intermediate undergo gentle phase catalyzed hydrolysis by attack of nucleophilic water at the anomeric center and form the product. Now let's come to the beta amylase. Beta amylase acts at the non-reducing chain in and liberate only beta maltose. During the ripening of the fruit, beta amylase breaks starch into the maltose, resulting in the sweet flavor of ripe fruit. Now let's come to the gamma amylase. Gamma amylase acts at the non-reducing chain in of amylose and amylopectin and liberate glucose. The gamma amylase has most acidic optimum pH of all enzymes because it active around at pH 3. It forms the amylose and amylopectin and gives the product glucose. Now let's come to the summary. Amylase have three types: alpha, beta, and gamma amylase. Where alpha amylase present in the saliva and pancreas, and the beta amylase present as the seeds and fruits of the plants. Gamma amylase present at the small intestine. Alpha amylase randomly cleave the alpha 1,4 glycosidic bond and beta cleave the second 1,4 glycosidic bond, whereas gamma amylase cleave the last 1,4 glycosidic bond. Alpha amylase give the product maltose and dextrin, whereas beta amylase give the product maltose. Gamma amylase give the product glucose. Thank you.